Here he comes.
So that's what happened. We are getting rid of that phone in 2 days, so that won't happen again. I'm sorry about that. It's irritating as all heck, but that means we're going to have 2 recordings of this in the memory. Okay, let's get back to so the conversion of serine to glycine provides the donation of the methylene group to tetrahydrofolate. Now this enzyme, serine hydroxymethyltransferase, is also highly conserved. It's a dimer in bacteria. In eukaryotes, it's a tetramer. It's a very interesting enzyme, frankly, biochemically. And it's a tetramer in humans. It's also a target for various types of anti-cancer drugs. Now that methylene group can be subject to modification of its oxidation by either reduction or oxidation. So there's a N5N10 methylene tetrahydrofolate reductase that will reduce the methylene tetrahydrofolate to a methyl tetrahydrofolate. This is an NADH requiring enzyme, and that converts it to the N5 methyl group. So that's methyl tetrahydrofolate. And tetrahydrofolate can also carry the single carbon group in the form of a formate. So it can carry it as an aldehyde. The N10 formal tetrahydrofolate is produced by N10 formal tetrahydrofolate synthetase. The production of the formal group requires the expenditure of an ATP. And the formal group is used in the synthesis of purines. It's very useful to put an aldehyde onto a structure that you want to make a ring from, because the aldehyde's very reactive and it will react with the amine to form a ring. And then it's also used in the formation of N-formal methionine, which is the starting amino acid in translation in bacteria. So this is a picture taken from your book. And it summarizes the different reactions. So here's the most reduced form up here. So this is the most reduced form here. Methyl tetrahydrofolate. You can take some reducing equivalents from this, convert it into the methylene group, or this reaction can go the other way. That's the N5N10 methyl tetrahydrofolate reductase. That gives you N5N10 methylene tetrahydrofolate. There's the methylene. You produce this by converting serine to glycine. And this, as you can see, has also a pyridoxal phosphate. This can then be converted also into the formal group by means of a N5N10 methylene tetrahydrofolate dehydrogenase. This reaction, as you can see, is reversible. That gives you the N5 formal. There's a cyclohydrolase that will convert this to a N10. And then you can turn it into the form amino group by virtue of a cyclodeaminase. And then if you want to release a formate, you can simply use a N10 formal tetrahydrofolate synthetase that will convert this, the formate, into the formal group. So there's several ways to get the formal group, but you'll notice the formal groups, it's going to take several reactions. So you can make it from the methylene, or you can make it from the glycine to serine to formate, so on and so forth. So a lot of these reactions are reversible. Another cofactor involved in amino acid degradation shows some structural similarity to tetrahydrofolate, and that's called tetrahydrobiopterin. This cofactor will participate in oxidation reactions and oxidation reactions alone. We will show you one particular reaction in the reactions to come where we use tetrahydrobiopterin. And to do that, we need to first talk about amino acid degradation. So amino acids are degraded using common pathways. If you have amino acids that are structurally similar, the cell doesn't design 4 different pathways to degrade 4 different amino acids that are structurally similar. It'll shunt those 4 or 5 or 2 or 3 amino acids into a common pathway. That way it has one common pathway and then branch points of those pathways. So this is really true right here. Groups of amino acids are degraded to similar metabolic intermediates by similar common pathways. That's the piece I need you to hang your hat on, common pathways. Mutations in genes that encode proteins involved in amino acid degradation can cause various human genetic disorders. If you can't degrade certain amino acids, intermediate products will build up. Those intermediate products may have deleterious effects on human metabolism. We talked about some of these already, these defects right here, where you have problems with urea synthesis. So you have defects in arginase, defects in arginosuccinase or arginosuccinate lyase, defects in carbamyl phosphate synthetase 1. And all of these have very similar phenotypes, very similar readouts, early death essentially. So argininemia, arginosuccinic acidemia, carbamyl phosphate synthetase 1 deficiency. All of those are going to affect the urea cycle. Defects in tyrosine degradation can lead to alkaptonuria. They can also lead to phenylketonuria, depending upon where the defect in tyrosine degradation tends to be. With this one, the defect's very early because you convert phenylalanine to tyrosine. This one's more severe than alkaptonuria. And then albinism results from a defect in the synthesis of melanin from tyrosine. That is an inability to create reactive species from tyrosine. Those enzymes are called tyrosinase. Defects in tyrosinase can cause albinism, where your skin pigment is completely white, and your hair, and your retinas, and so on and so forth. Homocystinuria is a defect in methionine degradation, because a defect in methionine degradation creates more homocysteine. The defective enzyme here is cystathionine beta synthase. Defects in the conversion of propionyl coenzyme A to succinyl coenzyme A due to deficiencies in methylmalonyl coenzyme A mutase have very severe consequences. That leads to so-called MMA, not mixed martial arts, but methylmalonic acidemia. And then maple syrup acid disease is due to defects in the inability to properly degrade these branched chain amino acids: isoleucine, valine, leucine. That's due to a defect in an enzyme that degrades all of these in their first step, branch chain alpha keto acid dehydrogenase. It's a complex. Defects in that complex cause maple syrup urine disease, very severe disease.
Is it called maple syrup urine because of the color or because of the smell?
Because of the smell, and because the urine becomes very thick with these keto acid derivatives that fill up your bloodstream. The kidneys work very hard to excrete them, and the urine is thick like maple syrup. That's why it's called maple syrup urine disease. All right, now here you can see these different amino acids are degraded into various end products. Some of these amino acids appear in multiple end products. That's due to the fact that the molecule gets cleaved, and one part of it gets degraded into one thing, another part of it gets degraded into another. So for example, phenylalanine here and tyrosine here, they get degraded into acetoacetyl coenzyme A. But another part of it also gets degraded into fumarate. And then isoleucine and threonine, these two here, they get degraded to acetyl coenzyme A, but another part of the molecule gets degraded to succinyl coenzyme A. So some of these do double duty, as you can see. Same thing here with tryptophan. And tryptophan over here. Part of tryptophan gets converted to pyruvate. Same thing with threonine. Threonine, you'll notice, ends up in several places because threonine is degraded by more than one pathway. That's the other thing. Different tissues in our body we use different pathways for some of these amino acids. That makes it kind of confusing. Some of these amino acids are degraded to intermediates that can be converted to glucose. So you see that in the rose color here. And that's your glucogenic substrates. Others are degraded into things that'll be used to make ketone bodies. Those are ketogenic. Some are a little bit country, a little bit rock and roll. So isoleucine is a glucogenic amino acid. Others are more ketogenic. So lysine is a ketogenic amino acid. Leucine is a ketogenic amino acid. Where these others, like threonine, tryptophan, they're all considered glucogenic. So true ketogenic amino acids are really few and far between. The rest are all glucogenic. So as we said, amino acid degradation occurs via rather common pathways. So the amino acids leucine and lysine and phenylalanine and tryptophan and tyrosine are all degraded to acetoacetyl coenzyme A, and acetoacetyl coenzyme A gets converted to acetyl coenzyme A. So these here, tryptophan, phenylalanine, tyrosine, and so on and so forth, they all get converted to acetoacetyl coenzyme. They all get converted to acetyl coenzyme A. Same with leucine, same with isoleucine. Except isoleucine, part of the molecule, and the phone's ringing again, which means I'm probably going to get cut off. Let me. There, can you still hear me?
Yes.
There, good. I shut it off in a hurry and that seemed to help. I put the phone right here so I won't have to get up anymore. I'm sorry about that. So what happens when you do this at home? All right, so essentially except for isoleucine, and you can see here, this carbon right here, this carbon right here, this carbon right here, these carbons here, they're the piece that's going to get converted into your propionyl coenzyme A. This piece over here gets converted to acetyl coenzyme A. And then that gets converted to succinyl coenzyme A, which gets fed into the Krebs cycle. So all these get converted really to acetyl coenzyme A. These four, right, well, these five right here, leucine, tyrosine, phenylalanine, lysine, tryptophan, get converted to acetyl coenzyme A via acetyl coenzyme A. Leucine gets converted more directly. We'll look at these pathways more in detail. I just wanted to give you the broad outlines first. Alanine, cysteine, glycine, serine, and tryptophan, part of tryptophan, they get converted to lysine. Excuse me, pyruvate. So with tryptophan, it's essentially this piece right here gets converted to pyruvate. And you can see here, you just get rid of this. You can take this off. You get alanine. You get rid of this, you have pyruvate. that's where your pyruvate comes from. The rest of this essentially, that's what's going to happen essentially here. And this shows that here. So this gets converted to alpha ketoadipate. And alpha ketoadipate is also the pathway that's used to degrade lysine. So tryptophan and lysine will share a degradational pathway. But this piece gets converted to pyruvate. Serine, as we said before, can go straight to pyruvate. Cysteine, in 2 steps, can go to pyruvate. So with serine, there's an enzyme called serine dehydratase. It dehydrates serine. It deaminates serine. Excuse me. And that yields pyruvate. Glycine is converted to serine via the enzyme we just saw before, serine hydroxymethyltransferase. Glycine also is degraded via the glycine cleavage enzyme. The glycine cleavage enzyme occurs in mitochondria. If glycine is subjected to the glycine cleavage enzyme, it does not contribute to either ketogenesis or glucogenesis. And in mammals, a lot of your dietary glycine gets converted to ammonium and carbon dioxide via the glycine cleavage enzyme. Threonine gets converted to the 2-amino 3-ketobutyrate by threonine dehydrogenase, and then that gets converted to glycine by a relatively complex reaction with a very interesting enzyme, 2-amino 3-ketobutyrate coenzyme A ligase, that we'll talk more about. That gives you glycine, which goes to serine, which goes to pyruvate. So again, common pathways, common pathways. And we could say also here that threonine gets converted to pyruvate as well. So threonine should be in here too. Succinyl coenzyme A is the product of the degradation of threonine and valine and methionine and isoleucine. So methionine is converted to homocysteine. Homocysteine is degraded to alpha ketobutyrate. Threonine is also converted to alpha ketobutyrate. Alpha ketobutyrate dehydrogenase will convert this to propionyl coenzyme A. That then undergoes the reactions of beta oxidation that eventually convert it to succinyl coenzyme A. Valine also gets converted to propionyl coenzyme A. So does isoleucine, but another part of isoleucine is going to be converted into acetyl coenzyme A, which you can see right here. And then mutations, as we saw in cystathionine beta synthase, cause homocystinuria. Glutamate is degraded to alpha ketoglutarate, as is glutamine, as is histidine, proline, and arginine. So these are the so-called glutamate family. And so histidine, arginine, proline, glutamine, all are converted to glutamate, and then asparagine and aspartate are all converted to oxaloacetate. This is just very few steps. No issue there. Aspartate is deaminated by aspartate aminotransferase to alpha ketoglutarate. The amino group gets transferred to some other alpha keto acid. Arginine gets converted to citrulline in bacteria by virtue of the enzyme arginine deaminase. This is a bacterial enzyme. You don't see this enzyme in eukaryotes very much. You see it in plants. It's not the main way you get rid of arginine, though. But this is an enzyme you don't see very much in animals. And that converts arginine to citrulline. All right, let's look a bit at the degradation of some of these amino acids. First, let's take a look at the catabolism of histidine. So histidine is converted to glutamate. First thing you need to do is you need to get rid of the imidazole ring. So you're going to remove a formamide group. That's going to require tetrahydrofolate. So here are the reactions by which you degrade histidine to glutamate. Glutamate is then converted to alpha ketoglutarate. So first, histidine is subjected to an enzyme called histidase. Histidase. Some books will call this histidine ammonium lyase. Probably a more common name is histidase. Patients who lack histidase suffer from a condition called histidinemia. Histidinemia. Histidine and its products will accumulate in blood plasma, urine, and in the cerebrospinal fluid. This condition, depending on its severity, can be completely benign or can cause speech defects and mental retardation. You see this condition in about 1 in 10,000 births. You can normalize people by restricting histidine consumption in the diet.
What foods commonly have histidine in them then?
Usually meat products. Typically meat products, although white fleshed meats will have less histidine. So your chickens, your fish, that sort of thing. Soy has less histidine, has a little, but not much. That will restrict histidine. When you get rid of the ammonium group there. You now create a double bond here. That creates this compound called urocanate. Urocanase will add a water across this, but it does something really funny. It gives reducing equivalence here, but the oxygen goes up here. And you get rid of this double bond here. You now make 4-imidazoline 3-propionate. And now you add a water across this as well. By adding a water, you're going to break this bond right here. And that's called imidazoline propionate hydrolase. And you get N-formamino L-glutamate. So you're going to get rid of the formamino group. And to do that, we take our old friend, tetrahydrofolate, and that is glutamate formamino transferase. The enzyme that regenerates this molecule, you might remember, requires vitamin B12. So if you have a vitamin B12 deficiency, you may suffer from high levels of your N-formamino L-glutamate. This is called FIGLU. Deficiencies in this hydrolase here it can also be caused if you have low folate in the diet. And in that case, you'll find high levels of this compound in the urine. The enzyme that regenerates this tetrahydrofolate is called cyclodeaminase. And then once you get rid of that formamino group, you convert this to glutamate, and then that's converted to alpha ketoglutarate by deamination of that amino group right there. That's how you degrade histidine. Okay, I need to. Oh, good gravy. Come on, computer. Work with me here. Nope, you're not working with me. Okay, can you see my blackboard here?
Yes.
Thank you. Everything's better with computers except when they don't work. Now, when you consider histidine, yeah. So that's the amino acid histidine. That's backwards. Now, how do I fix that? Oh, for pity's sake.
It looks like it's the right way for us.
Really? Looks
Yeah,
backwards to me. Okay, I'll keep going then. Now, there is an enzyme called histidine decarboxylase. This enzyme is ubiquitous, and that is going to generate and can you guess what that's called? That is called histamine. And that is precisely what causes you to go achoo, achoo, achoo during allergy season. So this is an important neurotransmitter, but it's also loaded up in mast cells. And when those mast cells degranulate, that's what essentially causes this binds to the H1 histamine receptors. And those H1 histamine receptors, when they're activated, that's what causes the effects of allergies. So histidine also gets converted into histamine. Okay. All right, let's take a look now at the, whoops. Computers, no, cat, I can't play with you now. Yes, Your Majesty. Yes, you're the queen, but I can't play with you. Now let's take a look at the degradation of these aromatic amino acids. Their degradation is quite complicated. They get degraded to fumarate, but the errors in this catabolism leads to genetic diseases that have been fairly well characterized. Oh, gosh, noisy. So interestingly, phenylalanine gets converted to tyrosine before it gets degraded. So tyrosine and phenylalanine share a common degradation pathway. Consequently, except for mutations that affect this particular enzyme here, phenylalanine hydroxylase, mutations that affect phenylalanine degradation will also affect tyrosine degradation as well. So let's go through these step by step. This is a very interesting pathway, but it's also a very complicated pathway. So the first step involves the conversion, but really it's a hydroxylation of phenylalanine to tyrosine. And it uses a mixed function oxidase called phenylalanine hydroxylase. And this enzyme uses tetrahydrobiopterin as a cofactor. And the NADH is to reduce the tetrahydrobiopterin. There's actually a tetrahydrobiopterin reductase enzyme that will reduce the tetrahydrobiopterin. That's why the NADH is included in here. So you end up with hydroxylated phenylalanine, which is otherwise known as tyrosine. Now, errors in the phenylalanine hydroxylase enzyme or errors in either tetrahydrobiopterin synthesis or tetrahydrobiopterin reduction will both lead to this condition, phenylketonuria, PKU. PKU, I realize, sounds like a university. It's not. It stands for phenylketonuria. And phenylketonuria nowadays is also known as hyperphenylalanemia. Hyperphenylalanemia. And that hyperphenylalanemia comes as a result of mutations in several different genes. So it could be due to mutation in phenylalanine hydroxylase. That was the original description of the regional phenylketonuria. Or it could be due to mutations in dihydrobiopterin reductase. Or it could even be due to mutations in genes involved with dihydrobiopterin synthesis. Those can also lead to hyperphenylalanemia. There's a test for this. It's a genetic test that you give to newborns because if they have hyperphenylalanemia, you need to restrict phenylalanine intake right away. If you do that, they can have normal brain development. If you don't, phenylalanine will cross the blood-brain barrier. It'll interfere with neurotransmitter synthesis, and it'll cause permanent mental retardation. So if this runs in your family, there's a good genetic test you can order and learn right away if the baby needs to be put on a restricted diet. So that's phenylalanine hydroxylase, uses molecular oxygen and dihydrobiopterin. And here's this pathway that you can see here where you have essentially the regeneration of your dihydrobiopterin. This is essentially where you use your NADH. This goes to the quinoid form, to the tetrahydra form. You can see the reduced form has your reducing equivalents here and also here. Whereas here and here. And then this essentially leads to the hydroxylation of the phenylalanine tyrosine. Now, once you make tyrosine, now you deaminate it to make p-hydroxyphenylpyruvate. P-hydroxyphenylpyruvate, both dietary tyrosine and glucocorticoids increase the synthesis of this enzyme. Mutations in tyrosine aminotransferase will cause a condition called tyrosinemia. And then the p-hydroxyphenylpyruvate will undergo a complex reaction that's catalyzed by a dioxygenase. So this very complex reaction involves a decarboxylation, oxidation, migration of the carbon chain, and a hydroxylation. It's a very, very complex reaction. The same enzyme catalyzes all four activities, and a cofactor for this enzyme is ascorbic acid or vitamin C. let's just say vit C. Tyrosinemia 3 and tyrosinemia 2 phenotypically are indistinguishable, except that in the case of tyrosinemia 3, p-hydroxyphenylpyruvate will be produced in the urine, whereas with tyrosinase 2, it will be tyrosine that you will excrete into the urine. So that makes this compound called homogentisate. Homogentisate. Homogentisate is subjected to another mixed function oxidase, homogentisate 1,2-dioxygenase. This will open the ring. And you get from this malonyl acetoacetate. And so this piece here is this here. And then this business here is essentially this right here. So this carbon right here, let's circle it here, is this carbon right here. So that's converted into a ketone from an alcohol. This double bond gets moved over here. And then this gets converted into a carboxylic acid here. Mutations in the gene that encodes this enzyme will cause homogentisate to build up in the body and to be excreted into the urine. Homogentisate, when it hits the air, turns dark. So in this case, the urine turns black. Homogentisate will also build up at joints and cause a late onset arthritis. This condition is called alkaptonuria. And in alkaptonuria, you will be able to detect homogentisate in the urine. You'll also have high homogentisate levels in blood. Malonyl acetoacetate is now the substrate for an isomerase. And that is going to take this cis situation here and convert it to a trans situation here. And you can see what's going to happen now. That's fumarate. This is acetoacetate. This is fumarate. You're just going to essentially cut this bond in half, and that's what's going to happen next. So this is an isomerase. You now make fumaryl acetoacetate, and then fumaryl acetoacetase, also known as fumaryl acetoacetate lyase or fumaryl acetoacetate hydrolase, will convert fumaryl acetoacetate to acetoacetate, and fumarate will then enter the Krebs cycle, and acetoacetate will then be converted to acetoacetyl coenzyme A, which will then be converted to acetyl coenzyme A. Mutations that affect the ability of cells to break down fumaryl acetoacetate will also cause tyrosinemia. Patients with tyrosinemia show symptoms that are similar to hyperphenylalaniemias. So early in life, these intermediates can cross the unformed blood-brain barrier and cause severe mental retardation. Restricting phenylalanine in the diet early on can prevent this from happening. Alternatively, an inability to degrade tyrosine so that it cross-links and forms melanin is due to a deficiency in an enzyme called tyrosinase. And that leads to albinism. Tyrosinase is a copper-containing enzyme, and that leads to albinism. And it looks to me like we're out of time. So we'll get to the branch chain amino acids next. As soon as I hear about my PCR test, I'll be in touch with you. For now, you guys know as much as I know. So I don't know if I told you I had a lateral flow antigen test this morning. That was negative, but I still have to have a PCR test. Until I have a negative PCR test, I can't go anywhere near campus. Those are the rules. I support the rules. So until now, till then, this is the way it be. When I know more, I'll let you know.
Did you still
So-
get your vaccine?
I got vaccinated.
I
I
was
vaccinated
hoping,
Friday, had a
right.
very sore arm.
I was
Friday night.
hoping. I was gonna say I was gonna be so sad if like you didn't get to get it
Oh,
because
basically I said I'm getting vaccinated come heck or high water.
right,
I didn't care.
right.
at
'Cause
that
you don't
point.
want to have to wait again because that's
Yeah, no, but my biopsy for my pinky had to be redone. So, oh well, can't have everything.
Mhm.
So yeah, I will keep you guys in the loop, and until then, send me emails if you have questions. I'll just be here, probably doing yard work and the occasional whatever. So, questions, anybody? No?
Nope.
Okay. Don't forget you have a quiz online. It's due tonight before midnight. So it's only 5 questions. So as the saying goes, get her done, get her done.
Thank you for the reminder.
Yeah.
Sounds
Okay. I will see you all later. Amanda, pet your kitty for me. Bye, gang.
okay. Bye.
Bye.